Maternal diabetes alters transcriptional programs in the developing embryo by
Actosine.
Hina Khan.
and Daishraj Bali. Introduction. The aim of this presentation is to present an overview of a research article written by Pavlinkova, Salbum and Kaplan entitled Maternal Diabetes Alters Transcriptional Programs in the Developing Embryo. Pavlinkova et al. Hypothesized that maternal diabetes may cause developmental defects caused by altered gene expression that consequently affects critical development pathways in the embryo. It has been found that maternal diabetes disrupts embryonic development and causes diabetic embryopathy such as cardiovascular malformations, neural tube defects, and congenital malformations. Several studies have identified a link between increased levels of oxidative and maternal diabetes. In contrast, other findings have found with the administration of antioxidants such as vitamin E and C reduced incidences of developmental defects observed from experimental models of intrauterine exposure to diabetes. Studies have shown that reduced expression of PAX3, a gene involved in neural tube defects, were found in embryos from diabetic mouse dams at gestation with neural tube defects evident by day 10. It has also been found that PAX3 deregulation may be the cause of oxidative stress, which consequently causes heart defects and involves neural crest cell derivatives. WNT is made of proteins that highly form conserved and secreted signaling molecules that regulate cell-to-cell interactions during embryogenesis. It has been found to be affected in the diabetic exposed mouse embryos. The evidence provided therefore supports the link between diabetic pregnancy and altered expression of key genes involved in development of the embryonic tissue. Altered transcriptional regulation has been implicated as a potential pathogenic mechanism in diabetic embryopathy. Diabetes were induced 7 to 9 weeks old female mice by 2 IP injections, streptozotocin, within a 1-week interval. Dams were set up for mating 7 days after the last injection, and the day of detection of a vaginal plug was counted as a day of gestation. Embryos from dams whose blood glucose levels exceeded 250 milligrams were used. RNA was isolated from embryos using Trizol, a reagent for the isolation of high-quality total RNA or the simultaneous isolation of RNA, DNA, and proteins from a variety of biological samples. 2 controls and 5 diabetes-exposed embryos were used. This was to identify and observe a range of phenotype spectrums of diabetes exposure in pregnancy. Of the 5 groups exposed embryos, 2 of them had neural defects. Experiment 1, samples were individually hybridized. Experiment 2, equal amounts of RNA prepared from 3 individual embryos were pooled into 1 sample. Each embryo was from a different pregnancy, and pools were constructed for a total of 9 control embryos and independently for 9 diabetes-exposed embryos. All embryos were morphologically normal. Expression levels present, marginal, and absent flags were determined with default parameters through comparison of matched and mismatched oligonucleotides for a respective genes sequence. Quantitative real-time PCR was performed as described on cDNA samples from individual diabetes-exposed embryos and controls. Normalization was done to polymerize epsilon cDNA in the same sample. Pol 4 levels were ineffective by the maternal diabetes on experiment 1 and experiment 2 arrays. Primers were positioned across exon-exon junctions to exclude amplification of potentially contaminating DNA. The amplification products were designed to originate from
Results. Overall results displayed show that sequence motifs in promoters of genes affected by diabetes suggest potential binding of transcription factors that are involved in responses to oxidative stress and hypoxia, which are 2 conditions that are known to be associated with diabetic pregnancies. A 6th of the deregulated genes have known developmental phenotypes shown by functional annotation in mouse mutants. Over 30% of the genes have identified transcription factors and chromatin-modifying proteins or components of signaling pathways that impact on transcription. Figure 1 shows experimental approaches to determine gene expression profiles of normal and diabetes-exposed embryos. Individual embryo samples were used in Experiment 1 and a pooling strategy was used in Experiment 2. In Experiment 1, expression profiles of 2 control and 5 diabetes-exposed embryos were surveyed. Panels A and B show the 2 independent microarray experiments. Panels C and D show expression profiles where each colored vertical line represents the expression signal for one gene and the row represents individual embryos. Red represents increased expression, blue reflects decreased expression, and intermediate colors represent minor changes. These graphic representations show that expression profiles for embryos exposed to maternal diabetes differ considerably from control unexposed embryo profiles.
Table 1 shows the genes that have been affected by maternal diabetes, which are classified by cellular function. 100 genes in total were affected by maternal diabetes out of a total of 126 genes. Control embryos exhibited profiles similar to each other, and differences to the expression profiles of individual diabetes-exposed embryos are visibly obvious. These results support the hypothesis that maternal diabetes affects gene expression in exposed embryo.
Table 2 shows validation of microarray results by quantitative RT-PCR.
High glucose levels cause malformation of the embryo in diabetic pregnancy. Excess glucose in experimental animals cause diamorphogenesis, which is a process of abnormal tissue formation of embryos in glucose-injected dams. The precise mechanisms by which the hyperglycemia induces diabetic embryopathy is not clear, although involvement of the GLUT2 transporter has been demonstrated. Genes which are functionally involved in responses and mechanisms of the effects of antioxidant treatments in diabetic embryopathy remains to be investigated. Similarly, folate supplementation has been shown to be protective against NTDs in diabetic pregnancies. We found transcription regulatory genes highly overrepresented among our diabetes-affected genes. For the majority of genes we tested, the independent assay confirms the initial finding that expression levels are altered in embryos exposed to maternal diabetes. Given the proposed role of oxidative stress and hypoxia in the pathogenesis of diabetic embryopathy, it's striking that 97% of the genes affected by maternal diabetes carry in the upstream regions potential binding sites for transcription factors that are involved in responses to oxidative stress and hypoxia. Neural tube defects are easily detectable at embryonic day 10, which was when the mouse embryos were isolated from diabetic or control dams.